So to start with chapter 19 from your textbook. Okay, chapter 19 from your textbook. Could you see here a bit of dark? I want to check it out. Okay. Could you clearly see the letters which I am writing it from Victoria? Yes?
It's small.
It is small, so you can't see that. Okay, I will make it a little bit bigger. Chapter 19, I'm writing it. Is that right? Red font? No? Could you hear? Could you see?
Yeah, it's better.
It's better now? Okay. I have the other
Can you
pen.
see it?
I didn't know where I kept it. So. Let me see. The light reactions of photosynthesis. That's what we are going to study today. Okay. This is the light reactions of otherwise the photosynthesis. Light reactions or photosynthesis. Okay. Without light, there is no life. Okay, light and life are integrated one. So to sustain the life, you need what? We need food, and for respiration, you need oxygen. Okay. For both, light energy converted to food energy or chemical energy, and this is the part how the light energy from the sunlight is going to form the food and for the respiration. So food to consume and oxygen to metabolize this food. So here I put an organism, consumer, I put it, consumer. And this one is the producers. And these producers use this light energy, and this light energy is coming from sunlight. So we are the other biological systems. We got the organism, I put, they are consuming it. At the same time, there is a part is being used, producers. And here, this one is mainly on the bacteria. They can also produce food, and as well as the microbes. I put microbes, use the sunlight, and also plants. So when you get this phenomena of how this light energy is being converted to food or starch and sugars and also liberation of oxygen. So that is the oxygen what we breathe in the planet Earth. Okay, and the food whatever you eat, either we are eating directly plant derived food, or we are eating meat, which is the animals, and that consume the plant in an indirect way. So in either way, the life on this earth is depending upon the light. So that's what my point. And it is not like life back to the light. So that is, we are deriving, we are getting the energy, one form of energy, and then we are using that energy for our purposes. So how this light is trapped, in other words, how the light is trapped. And then how this light is being, the trapped energy or the trapped light is stored in what form? So these are the items that we need to study. At the earlier classes, what we did, most of this, the energy, whatever we talked about energy, and that energy like ATP and and then creatine phosphate or high energy bond and and some of them they are also heat energy. You might have studied on that. So all of them which we have done before in the previous class. So today we are going to study about how the light is trapped and stored in what form. Okay. So as I mentioned earlier, the microorganisms, which is mainly on algae. Okay. Algae and bacteria, algae and bacteria, they do. The algae, they form around 60% of the conversion of the light energy. 60%.
Yeah,
Oh. You can't see that?
Yeah, the one that you're using is black or blue?
Yeah, blue.
It's very hard to see.
You got to see? Okay. Right. Yeah. I'll just check that one here. Okay. So the conversion done by these algae, that means the green plants. Green plants, they do. The algae. One example.
The red doesn't look good.
Red is not good.
The other.
The black one.
The black one, yeah.
You do that? This one okay?
Yeah, that one.
Okay. So when you see the sky on the earth, you have a blue sky, right? Yeah. If the blue sky, let me see. Could you see now? No? Is it better?
Yeah.
Oh, wonderful. Okay. Is the blue sky, how do you get this blue sky? What is the blue sky looks like when you come to the earth?
Is it all of them? Or is it
Yeah, algae. I mean, the Earth, the planet Earth, they contain a large amount of the sea, right? We how much? 1/3 is the land and 3/4 is the sea, right? Sea water. That sea contains the algae. There are a lot of that actually reflect onto getting onto the water. Is getting vapors, so the algae spores and everything is getting onto the sky. That constitute actually the blue sky. We have it. If we don't have a pure air or something like, we don't have the sky. So if there's a blue sky, which is any other planet, that means there's a lot of chances of getting of water as well as the organism, as well as the life now. But we didn't find any other planet on solar system, no. So, coming up to our point, without the transformation of life, without this transformation, the light to the life, we cannot exist. The life won't, there's no life at all without plant. So that's the ultimate thing. So the process of converting the light, what is the light now? I just go into one by one. Light.
We can't really see what you're writing.
Here, okay, that's a little bit of thing. Let me write again on this one. Could you see now here, light? A little better? Let's, yeah? No? Is it okay? Could you see? Or you want
Not really.
You want the lights now? I don't know why this is getting it some. I just give you the contrast on that one. No? L-I-G-H-T, light. No, I want to, yeah. I will write large letters probably, if you can, L-I-G-H-T. Could you see now? The large one, larger font?
Yes.
Oh, okay, so I will write large, then you can get it out, not with the small one. Okay. So the light energy, the process is nothing but electromagnetic. Waves or electromagnetic waves are what? Waves are radiation. You see now in here? Okay. So electromagnetic radiations. So if you do the electromagnetic waves or radiation, this is the one, I call it is the light. But you know the light is having its own visible light and invisible one, ultraviolet. And so visible range is, we call it a VIBGR. Do you know about VIBGR? V for violet, I for indigo, B for blue, G for green, Y for yellow, O for orange, and red, R for red. So all these 1, 2, 3, 4, 5, 6, 7 colors. What the 7 colors? 7 colors, that is the one where you have the light. The light on constitute of the 7 different wavelengths. So when you see the light, sunlight, it comes out from the, like a rainbow, right? So you have a different color, meaning different radiation. Violet is one particular wavelength, and this is one wavelength. Blue is another wavelength. Green is another wavelength. Yellow is another. So you have 7 different wavelengths of light which is coming. That meaning electromagnetic waves. So not one wave. There are 7 different waves, and also additional waves like microwaves and a lot of waves there. But I am giving here is only the visible range of light, visible range of the color, visible range of the electromagnetic waves which is coming up. So this light, suppose in some part of this light, it's been absorbed. I'll write it here. Absorbed, okay? Absorbed by pigments. And energy is stored, or light energy is stored. Okay. So now, what is the pigment? I mean, how, in what form this energy is stored? That is the purpose now of this class today. Okay. See, the light energy, as I mentioned earlier, I put light means always you imagine there's light energy, okay? Light energy. Okay. And there is the phenomena, we call it as a photosynthesis, where the atmospheric carbon dioxide and water, H2O, okay, they form 2 carbohydrates. Carbohydrates. Carbon, hydration is water. So hydration of this carbon we call it carbohydrate. Okay. So this is coming from your atmospheric air, and this is again from the ground. So there must be some structure, or the organisms, they have to have capturing this light energy. That is again in a plant leaf, and in the leaf there must be a pigment. The pigment normally we call it as chloroplast. Is an organelle like mitochondria, cellular organelle. The chloroplast contains chlorophyll as a molecule or a pigment, chlorophyll. And this is as a green in color. Okay. So that's why the plant leaves is a green in color. So you have to have the green color of the leaf and the pigment chlorophyll, and that is going to, the reaction is here, and the leaf is going to capture the light energy. So these are the scenario. Something like a drama, a light source, and then must be a stage, and the stage is going to be the leaf, and the actors here, which is coming on the chlorophyll, carbon dioxide, and water. Okay, and these are the actors, and they all produce a drama and then produce say carbohydrates. So just think in that way. So everything is taking place in a one particular fashion. Okay, now let's go into the next one, the range. During the conversion, like a carbon dioxide plus water, okay, H2O, you have a light. So if you CH2O. I put CH2O plus O2. Okay, 2 oxygen here and 1 oxygen 3, and then here 2 oxygen 1 3, and then C and then H2 CH2. So this CH2O, this one is we call it as a short starch unit or carbohydrate unit of that one. Carbon, hydrogen, oxygen. That's basic unit. And oxygen is evolved as a side product, as a side effect. Otherwise, when you synthesize the starch, the oxygen is released into the environment, and this oxygen we can I mean, that's been used for the breathing of the organisms, for the aerobic in the air, oxygen. So if you don't have the plant, no plant, you don't get oxygen to breathe. So that's what we have to, aware of the plant and not deforestation. Reforestation is important on that part. Okay. The carbon, see the carbon dioxide from the air, and this is the water vapor. Again, you can have the liquid. Okay. Now it produces a solid on that part. So if you think on the status of the material from air and water mixed together and carbohydrates. Yes, air is there, water is also there in the air, but why don't they synthesize starch and oxygen in presence of sunlight? But everything is there, but you need to have a plant or other organism, an industry, which will use this molecule and use this light energy to convert into carbohydrates. So we are going to study now about the photosynthetic organism, about the plant. Okay, the photosynthetic organisms, we call it as photosynthetic organisms. Photosynthetic organisms, they are called by name autotrophs. A-U-T-O, autotrophs, T-R-O-P-H-S-S, P-H-S, autotrophs. Meaning they are self-feeders. Self-feeders. Okay. So the another type of organism which will use this phenomena, which is called the heterotrophs. Okay. That is, the organisms obtained energy from chemicals as a fuel, and they are called as heterotrophs. But these chemical fuels, where they will get normally, the chemical fuels again is coming from the autotrophs or the photosynthetic organisms. They will produce more of the carbohydrates and proteins and fat and all. So then the heterotrophs will use these autotrophs products, and and these organisms we call it heterotrophs. Why these autotrophs? The plant themselves will synthesize the food, and they will also use the same food. So that's why you call self-feeders. They are not depending upon anybody else. They depend upon them only because they are producing the food, and they are using their food if they want to use it. So that's the kind of stuff which we need to understand, the two different one. And now the importance of the energy, solar energy, we call it as base. Okay, what is base here? Is biological application of solar energy. B-A-S-E, base. Biological application of solar energy. So this phenomena is very important in environmental point of view. As well as they can also produce more of applied the solar energy to synthesize a chemical fuel, something like a starch, and then from the starch you can convert into the chemical industries or glucose into biofuel. Biofuel, just for your car, an automobile. So all of them is directly depending upon the harvesting of solar light or solar energy into biologically application for food or fuel or any other form, even for the manufacture of wood or furniture, you name it. I mean, all the plant biology students, you might know about the application of the plant or some drugs, so everything. So the photosynthesis, in general, photosynthesis is the two part of this one, photosynthesis. Okay, two reactions it comprises of. One, light reaction, and another one is dark reaction. Okay. The light reaction, the light energy is transformed into two forms of biochemical energy. The light energy is transformed into two forms of biochemical energy. Biochem energy. Okay. Number one, that is a NADPH. Okay. And this is a reducing power. And then B is ATP molecule, which already you know about. So these two in the light reaction. That's what we are going to study today. And what happened in the dark reaction? Here you have a reduction of carbon dioxide. Okay, that means addition of hydrogen. In light reaction, you produce NADPH, where more of hydrogen is produced in the light reaction, and this hydrogen will use the reducing power will use here for the addition into the carbon dioxide from the air. When they plant, they use the carbon dioxide, and the light reaction produce more of hydrogen. That hydrogen is used for the carbon dioxide and produce of starch or sugar. And this dark reaction, in other words, they are called as Calvin cycle. Calvin, Calvin cycle. And this is the one, chapter 20, which we are going to cover next week of that one. Okay. So don't worry about this dark reaction. We will study about the light reaction today. That's what. Okay. As I mentioned earlier, chapter 17 and 18, which we have studied on respiration, okay, respiration, where we did study oxidation of glucose, okay, and also reduction of oxygen by electron transport chain. Here you have a glycolysis, which we did study, and then TCA cycle we did study, and the electron transport chain. Electron transport chain, and the electron transport chain is using reduction of oxygen, and thereby it's converted into water here, and the oxidation of glucose into carbon dioxide. So you have water. So this is purely of respiration. Okay, meaning if you reverse this phenomena of carbon dioxide and water to back into the carbohydrate, so that phenomena is just, I'm going to write it in a reversal order, CO2 plus H2O to produce glucose. This is the reversal of glycolysis, in a sense, the reversal of the catabolism or the anabolism because of synthesis of glucose. And it is not gluconeogenesis, but as you mentioned, this process we need to add more of energy. So you have to add energy. This energy is coming from light. That means photosynthesis. So in the earlier class we did study this. This is for the respiration and the oxidation reduction. And now this carbon dioxide we eliminated into the air by exhale on that breath. It's coming out if you breathe heavily, and then you get the expel the carbon dioxide outside the air. And the plant will use that carbon dioxide in presence of the light. You convert it into the carbohydrates and the glucose. So the phenomena here, you also have electron transport chain, and here also you have a mini electron transport chain, ETC. So the transfer of the electrons from light, the light is it will produce only the energy. It is not supplying of electrons. The electrons are here in one place. That's what most of the people, they don't understand. But I put it over here. Okay, here this box. Okay, here you have a light only. Light only is coming up. Okay, and it will produce more of CO2 and H2O, which is coming out. So this is the box or the universe or the earth or the people or the organisms are living here. So the light energy, they use only the energy which is coming out, but here they are converted to by bacteria or plant, and every organism, they use the light energy converted into the food and carbohydrates, and other organisms use this food and eliminate here. So it is going as a cycle, which is again and again. But the major fact is the energy is being used. Energy, light energy is being used here, stored and saved, and then work. For the work, we need this biological work or whatever mechanical work. We need the energy. So this energy is through food, and you have the energy, and then the waste product as a carbon dioxide, and then again this carbon dioxide is captured by the plant. So it is something like a cycle, carbon cycle. In other words, they used to call it. One is cellular respiration, another one is the photosynthesis. Okay. So they are chemically opposite. This is also electron, and here also using electrons is coming up the energy. So in other words, you have to get this energy is conserved in the form of food. So we will see how these electrons being transferred from one component into the other. So that's what we did last time about in the respiration. The generation of high energy electrons, as I mentioned before, the electron transport, right? Generation of high energy electrons. In chemistry class, you might have known about the electrons. Suppose it is the nucleus, neutrons, and then surrounded by different orbital of electrons, and electrons, s orbital, p orbital, sp. You know about the s orbital, p orbital, q orbital, d orbital. Good. You remember, right? d orbital, s orbital, p orbital. So the electrons are moving around, right? If I just put the electrons one direction, because the pair of electrons move opposite, right? So one electron going over there, one electron is going over there, one electron is going over there, and one electron is going up. So when you see these are the electrons' movement, the electron is not like a particle, but they have the force. They have got the negative charge on their own, and the proton or the nucleon, they have a positive charge. So in any atom, in the structure of the atom, and whenever the light which is falling onto the electrons, and the light is already mentioned before, they have electromagnetic waves, right? I like the electromagnetic waves, or energy, electromagnetic energy or waves. And these waves, these electrons also an affinity towards that, this magnetic force, so it will absorb this light energy or the electromagnetic. And then once it absorbs that one, these electrons will be excited. Will be excited. And when they excited in the single pair of electrons, and then it may either be coming to the ground, they call it as a ground state. I will write it in a separate fashion. Then, we are going to discuss now on that. I think in your book also they are given. Suppose if you have a electrons pair like this, and then you have another electron, another neighboring one, another molecule. If the light is flashing on this, one electron is being excited, which is being, is going up. Another state, excited state, it just move when in one pair, it will move from that. Why it will move? Because it has some energy, but it absorbs, the electrons absorb and then move. And then it comes down to the ground state. This is the ground state, and this is the excited state. G for ground, E for excited state, of the same atom. Some case, if it returns over here, that will be fine. Sometimes it may go into the neighboring molecule where it can go over there. This is another molecule just sitting over there, molecule 1 and molecule 2. So instead of coming here on the same atom or same molecule, it may go over here, the next one. And what will happen, the next one will get the energized state. So one particular molecule receives the energy, but when the electrons are moving from one to the neighboring one, so what you get, the neighboring molecule get excited. So the same one, when they move from one state to the other, the charges will be exchanging. Okay, because if you lose this particular atom, you lose one electron because of the electronic energy or electromagnetic energy or light energy push this electron from this atom to this atom. And thereby, what will happen to these charges become positive charge of this particular atom here. But this molecule, gaining of this electron, become more of the negative charge. So what you get, initially they are neutral, but once the electrical energy or electromagnetic light energy enter to the molecule, the electrons are excited, and that is returned. And when they return to the ground state, then absolutely no problem. But if the excited state to the next neighboring one, excited state, and thereby what will happen, the electron make the energy, light energy, make a neutral molecule into a charged molecule. So initially it is neutral, but now because of the electrical or the light energy, it is being charged. So there are some charged molecules which is used to capture the electrons and then moving to one atom to one molecule into the other, and this is the reaction. We call it as a reaction center. Reaction center. Okay. So now I explain how a molecule is being excited by the electron and then it is being moved. When previous case, that's on in a respiration electron transport chain, you have got the protons that is moving by enzymes and moving. There's no light is involved in that one. But here, the light energy is there. So we have a specialized compound for this, the molecule. We are going to see today. And then how these electrons are moving, how that will help to induce of, convert the carbon dioxide into and water to the carbohydrates. Okay. So when they move down the electron transport chain, as I mentioned earlier, it will produce as a charged molecule as a proton motive force. Otherwise PMF, proton motive force. And this is the proton motive force use ATP synthase enzyme to produce more amount of ATP. Okay. Just I'll explain to you. So when these electrons are moving over here, excited state, when they are moving into one to compound, one molecule into the other, when you get, then you are producing a large amount of proton motive force at that time. So that will help the ATP synthase, the proton motive force, push or activate the forces necessary, PMF is necessary to produce ATP. Okay? So thereby ATP is being synthesized. And once the ATP is synthesized, okay, in the synthesis, and this will be used for, this will be used for the synthesis of glucose. Okay. So in the high energy electrons, to synthesize the glucose from the carbon dioxide, you need what? The final conclusion to this, you need high energy electrons. So the high energy electrons, where do you get the high energy electrons? That is exciting by the excited state by the sunlight. Okay, and that will produce proton motive force, ATP synthase, and ATP is produced, and that is glucose. Now, how the regulation of the electrons is being produced, high electron. As I mentioned earlier, when the light is there and you get in the low energy state, and then it will become into the electrons. Electrons are moving to boost, right, to high energy state, high energy, because it absorbs that energy, microwave energy. The electromagnetic high energy electrons. And these high energy electrons are highly unstable. They are not stable. Immediately, when you give a person normally, he's okay, and all of a sudden he became a bumper lottery of $83 million on a one night, on morning, and what will happen? He got all of a sudden become excited, right? So not only him, but what will happen? The neighbors and everyone who heard the news, or his family members or friends, and everybody will be excited over there, more than him. The same way, if these electrons are getting a dose of energy, it's become highly activated, high energy electrons, and they are so unstable, and they want to return to the normal state or the ground state, so that they can stable. So at that time, they may enter into releasing the energy. There are two ways they will do. Okay, so I'll write it here. Unstable. They are unstable but high energy, right? High energy state. So when they come down to low energy state, they release in the form of heat, the energy. So how do you get this unstable energy? By light. So light is cold. It's not hot. It's not heat. But light energy. But this light energy now has been captured by the molecules in the leaf and chloroplast, chlorophyll. We are going to see that what type of molecule it is involved. And that one, which is going to be the highly energy state, and that high energy state is going to unstable, and that return to the lower energy state, and you get the energy, whatever captured is be released as a heat in a normal phenomenon. So if it is not and coming to the low energy state, okay, coming to the low energy state, the same molecule, it releases heat. But instead of releasing the heat, but this electron can also move to the neighboring molecule. That's what I explained a bit earlier. Neighboring molecule. And thereby make the other molecule bit excited. So like that, it's a chain reaction. One excitement to the another excitement, and that's a chain reaction going on. Okay, that's the excited electron. So what is the purpose of this excited electron? Excited electrons. highly excited electron. What is the purpose of these excited electrons? They produce the reducing power. Reducing power. The excited electrons help to produce the reducing power. In other words, they produce NADPH. It will produce more of NADPH. And the excited electrons indirectly, they also produce the proton motive force. Other words, you got PMF, proton motive force, and that will also use to synthesis to ATP, which I just mentioned earlier. Proton motive force will help to produce ATP. So these are the reactions, the light reactions, which we did study a bit earlier. The individual component, that's what I want to explain to you. The reaction systems is the photosynthesis. In a green plant, there are two systems in the light reactions. Light reaction, there are two systems involved. A, photosystem 1. Photosystem 1. And then B, photosystem 2. Okay. So we will see what happened in the photosystem 1 and what happened in the photosystem 2. In the photosystem 1, it will produce more of reducing power. I will write 1. It will produce PS. I put PS for P for photo, S for system. So photosystem 1, and this system in the chloroplast, it will generate reducing power. Reducing power, meaning it will produce more of NADPH, photosystem 1. And then another one, PS2 or photosystem 2, and this will use the oxidizes, photosystem 2 oxidizes water, see split water splitting, okay, and transfer the electrons to the photosystem 1, okay. So when the photosystem 1 generate the reducing power, it will, what is the generating power of NADPH? It will produce more of NADPH, meaning more of excited electrons, E for electron. It will produce more of electrons, okay, and push these electrons to synthesize the NADPH. So when you push this electron, as I mentioned earlier, when the molecule, as I mentioned, the excited one, and is true, and then it's going to the next molecule, and then to the next molecule, and then go to the next molecule. So when you do this, you are continuously producing positive charge molecules over here. Do you understand that? When the electrons are pushing, every time when a light energy is coming out, it pushes the next one, and then another light, it will push the another one. So every time when you move, what will happen? You are making this molecule more charged molecules over there, and this charged are what you have to replenish more of the electrons back onto the normal one, and this is being happened by photosystem 1, PS1, okay. And what photosystem 2 will do, it will oxidize water, H2O. It split the water and produce more electrons. It will produce more electron and supply the electron to photosystem 1. It will supply. So it is something like a coordination of photosystem 1 will produce more of electron to produce NADPH, and photosystem 2 will split water and it will produce electrons and supply the electrons over here, so that this will become neutralized. So you will get more electrons, which is coming in, and that is supplied by the photosystem 2. So in that way, I'll write it separately. Photosystem 1, it will donate more electrons, and then what will happen? The electrons are going away from that one, and the photosystem 2 will use the H2O, split the H2O, and then it will produce more of electrons and supply to this. So electrons is being supplied. So this is being used for the NADPH. More of electron power. So this is necessary. This is the product. Photosystem 2, to also use the light energy to split water and to produce more of electrons and also oxygen as a byproduct. That oxygen is being released into the air, and the electrons are donated to photosystem 1, and thereby you will have more of NADPH will be used. So water is used by the photosystem 2 and produce oxygen and electrons, and photosystem 1 also using the light energy and push this to synthesize the NADPH. Now, we used already water, and we also used the energy from light source with 2 systems and NADPH, and what is left over? Carbon dioxide. The carbon dioxide, that is going to be reduced using this phenomena, and that is during the dark reaction. That is chapter 20. We are not going to deal with that right now. But I want to confirm with you, are you understanding the photosystem 1 and photosystem 2? How the 2 systems are coordinating with the light energy. That's what I want to know. You follow? Any questions from this photosystem? Because I'm just going in deeper in this. Once you understand what's happening in photosystem 2, photosystem 1, what is the purpose, what is the use, and what is the product of these 2 systems, then the next step will be easier. Do you follow now? We are talking about photosynthesis. I'm not talking about photosynthesis at all. So we are going somewhere else. Yeah, that's why you don't think that any middle school students can do carbon dioxide, water will produce stars. That's not. We are not dealing with that one now, because you know already. Our main aim is how that phenomena occur by intermediate metabolism. That's what I'm saying. Don't have to expect me to write the equation of starch synthesis. No chance. It's not like undergraduate plant biology class. No, it is not. So we have to go in a little bit in a different way, in a molecular aspect. I mean, how actually it is photobiology, exactly one particular course, but we are teaching in one particular hour. I mean, one lecture course. But the light energy is being used to both systems, photosystem 1, photosystem 2, and thereby, I mean, different phenomena like NADPH is being synthesized, and then water is splitting over there. You follow now? So I'll go into the next one. Victoria, do you have any question? Could you see?
Yes, we can see.
Okay, great. So the photosystem, I talked about the photosystem 1 and photosystem 2. I talked about the electrons. How these electrons are moving from one to the other, that is again, as I mentioned before, ETC, electron transport chain, which we have studied earlier for the respiratory chain or oxidation of glycolysis pathway, and also in mitochondrial electron transport chain. And this electron transport chain that drives the force or proton motive force to produce the ADP plus Pi to produce ATP molecule. So this process in chloroplast, which is occurring, this electron transport chain which is occurring, all these phenomena, is a double membrane, double membrane organelle. I'm just giving an example of chloroplast. So this is an organelle, subcellular organelle, which is present inside the plant, and the chloroplast is the factory, which will harvest the light energy, uses the water molecule and carbon dioxide, and synthesize the starch. So how big this size of the chloroplast is the 5 micrometer in long, I mean the length. Let me write separately. 5 micrometer meter in length or long, or this one. Okay, this is the chloroplast. And it contains, there are the membranes, there are three type of membranes they have it. They have a outer membrane, okay, and an inner membrane. And this inner membrane, in other words, it is also intervening the intermembrane space, okay, and they also call the space, which we call it as a stroma, S-T-R-O-M-A, stroma. And this is the outer membrane space, inner membrane space, and then this inner membrane space is also called stroma. And also you have another side of the reaction. I mean, all this reaction which is occurring, which is in the stroma, that's a thylakoid membrane, like a membrane. I'll show you in a picture 19.2, and they have like a the stroma, the inside the stroma, this space where you get a dark reaction which is occurring, whereas the membrane, and then they also get the outer membrane. There you have a chlorophyll and thylakoid membranes, and there you have a light reaction. You will see in the picture, which I'll show you clearly. Okay. Now you just understand the membrane structure of the chloroplast. In stroma, we can observe the membranes and structure we call a thylakoid. As I mentioned, it's just a large inner membrane. This is the outer membrane, and you have got a piling of thylakoid membranes. They call the piling of grana, and then these are, each one is the granum. They have plenty of thing grana, and they are being connected by another membrane. So these are called thylakoid. They're called like thylakoid, and the membrane they call thylakoid membrane, and they have a thylakoid one unit we call it as a granum, G-R-A-N-U-M, granum. So you have many granum, so we call it a plural, we call it grana. So one granum, another granum, another granum. So when they linked together, we call it as a grana. So unlike mitochondria, in mitochondria, I'll draw like this, like an outer membrane, and then inner membrane will go like this. Right. So you have a cristae and matrix. That's what we call it in mitochondria. But in the chloroplast, you have the thylakoid membrane over here. These bridges from one granum and to another granum, they are called stroma lamellae, L-A-M-E-L-L-A-E, stroma lamellae. They are connecting to each other. What is the bridges? What is the thylakoid membrane? What they are made up of? And what these membrane spaces? They are the components of the chloroplast, and where the photosynthesis is occurring, or capturing of electrons. So these thylakoid membranes, they are, what is that thylakoid membrane? They are budding from inner membrane of chloroplast, inner membrane, I-N-N-E-R, inner membrane of chloroplast, just like a cristae of the mitochondria. And the thylakoids, they have thylakoids, they have the electron transport chain, or the component, okay? And the electron transport is occurring, and during the light reaction which is occurring, light reaction. Whenever I say light reactions, you should remember the photosystem 1 and what? Photosystem 2. They are there, reaction centers, they have it, okay? And they also produce the electron transport chain, they also produce what? Proton motive force, PMF, which will produce more of the ATP, synthase to ATP reaction, okay? The reaction centers it is electron transport chain and ATP synthase, and all of them they need light. That's what it is, a light reaction. Light reaction or light harvesting proteins. Light harvesting proteins are involved in trapping the light energy, and these light harvesting proteins, they are present in reaction centers. Reaction centers. What are reaction centers, which I explained earlier? Reaction centers where the electrons are getting excited and the electrons are moving from one molecule to the other molecule. Okay. And they also have the thylakoid membrane. They also have lipids as well as proteins as a components, so the membrane components. Okay, they are the membrane components. Membrane, thylakoid membrane components. Okay. Now, the plant cell, plant leaf. Okay, I'll just the next one. As a plant leaf, how many chlorophylls there? The chloroplast, they have between 1 and 100 chloroplast. The leaf they contain. That depending upon the species of the plant, you may have more or less of the chloroplast. Okay. What this chloroplast, where this chloroplast is coming from on the evolution point of view? They have like a endosymbiotic event. There are in a evolutionary process, they form as a endosymbiotic. Symbiotic. ENDOSYMBIOTIC. Endosymbiotic event occur. Suppose in the evolutionary process, when an organism, especially on a cyanobacterium, a bacteria can enter into the multicellular organism, something like a unicellular organism, like a bacteria, and this bacteria can synthesize its own autonomous body. That meaning it can reproduce, and it can also get the light, using the light, and it can produce its own food like that. Later on, these unicellular organisms is getting into the plant cell, and the plant cells, these are these organisms, and these plant cells inside that, and this unicellular bacteria, cyanobacteria, in the evolutionary process, it will become chloroplast. So originally, what is the chloroplast? Like in a bacterial origin. So that's what is the evolutionary process. The same thing is mitochondria, as well as for use of oxygen consumption, how to derive energy. So they also, the same endosymbiotic relationship is there, and they can also produce more amount of this energy as mitochondria in the multicellular organism. And this is one other thing, is what type of microorganisms you can verify in 19.4 in your textbook pictures, and the higher plants in the chloroplast, the endosymbiotic relationship, and that will be useful to understand where the chloroplast is coming from to the plant. Okay, endosymbiotic. Okay, what methods is being transferred in the chlorophyll molecule as a pigments? They have the electron transfer mechanism, and the component of the molecule, as well as the what type of chemical which is involved and the transfer of the electrons, we will see in a couple of minutes, and I will give you a break, and please come back maybe another 10 minutes, and we will go ahead and finish that. The photosystem, they have the absorption of light by photoreceptor molecule. Okay, so the light absorption by chlorophyll. chlorophyll molecule. The chlorophyll molecule, as you know very well, it is could you see down here? Light absorption by chlorophyll molecule, and this is the photoreceptor molecule. It's a green pigment, okay, and they have different types of chlorophyll. So one of the type is the chlorophyll A, CHL I abbreviate for a chlorophyll, and A is a type. Chlorophyll A is a type of compound, which is the receptors, which is photoreceptors. And this chlorophyll A, what is the chlorophyll molecule is made up of, or chlorophyll pigment? In other words, the pigment. You know the green pigment in the plant leaves, what is that one now? We are going to search into the chemical phase, and that is called tetrapyrrole, Pyrrole, tetrapyrrole, and they are substituted tetrapyrrole. They are substituted molecule. Substituted, meaning there are other compound is attached to the tetrapyrrole ring or tetrapyrrole molecule. I'll just show you the structure. In your textbook is 19.5. I will walk you through when I show the slideshow to you. In that structure of the tetrapyrrole, there is a nitrogen molecule or nitrogen atom surrounding the pyrrole. That is coordinated with the magnesium ion. Like this is the magnesium ion, and this is being coordinated. This is the nitrogen group, another nitrogen, another nitrogen, another nitrogen. Okay, and then you have the other atoms, other molecules are going through that one here. They are attached to it. Okay, I am not drawing it in this one, but I will show you. But this is the core structure of this chlorophyll molecule. So whenever, if you see a lawn, there is no green lawn, so what a gardener recommend, you have to add some magnesium compounds to your soil, and thereby your lawn looks more green. And then ferrin, iron, and magnesium in a particular proportion is lacking in the soil, so your lawn is look pale green or yellow in color. So you need to have more of magnesium into it. That produces this more of chlorophyll pigment, require more of magnesium, and thereby you get more synthesis of chlorophyll, and thereby your lawn become more green. So that is one of the a small clue. So the excited electrons are present in the pyrrole ring. Okay, I'll just write the chlorophyll A molecule. Okay, they have a reduced pyrrole ring. Reduced pyrrole ring. Pyrrole or pyrrole ring. Okay, whatever you call it. That pyrrole ring, in addition to the 5-carbon ring, another one, 5-carbon ring is fused. 5-carbon ring is fused to this pyrrole ring, reduced pyrrole ring. And also, the chlorophyll molecule, they also contain phytol. Okay, chlorophyll molecule also contain CHL. Chlorophyll molecule, they also contain PHYTOL. Phytol is another compound, and this phytol, 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 is a highly hydrophobic. Hydrophobic means water hate. Hydrophilic means water loving. So it is the non-polar compound. You know, hydrophobic, highly hydrophobic. They have a 20 carbon atom. 20 carbon unit, 20 carbon alcohol. They are esterified with an acid. In acid and alcohol we call it ester, right? The alcohol plus an acid, and they have a 20 carbon alcohol with acid, it form an ester. And this is the one which is attached to the phytol. This compound is attached to the chlorophyll molecule. Okay, it is the chlorophyll contain the 20 carbon alcohol acid ester, and chlorophyll molecule also having the pyrrole ring, and another one the chlorophyll contains, because of the the substituted with a conjugated double bond structures. Okay, they have conjugated. Okay, chlorophyll, again chlorophyll molecule structure. Okay, I said one side you have a pyrrole ring, and then another one is the phytol. Another one you have a conjugated double bond, D-O-U-B-L-E, double bond. What is the conjugated double bonds? See, if you have the structure of suppose is the C double. This C is a carbon, and then what I am writing it here, you can see single bond, okay, and this is a double bond, single bond, a double bond, a single bond, a double bond, a single bond, a double bond. So what you have alternate double bond and single bond, and they are called the conjugated double bonds. Do you follow what is conjugated double bond? So the chlorophyll molecule contains the conjugated double bond, and chlorophyll molecule they also phytol, they are alcohol 20 carbon unit alcohol with acid as an ester, and also they have a pyrrole ring, all of them. So this double bond, normally in chemical name or the name nomenclature, we call it as a conjugated polyene, P-O-L-Y-E-N-E. E-N-E means double bond, in the chemistry. Conjugated polyene. This conjugated polyene, the polyene means the double bond is something like an electrons, right? These are the electrons, this is the one electron, but here the cloud of electrons. So that's why the bond formation from one carbon to the other. And the carbon of this polyene, 1, 2, 3, you have a series of this polyene, and the electrons are not belong to one particular nucleus. I will draw like this, okay, just for your understanding. Double bond, single bond, double bond, okay. So and here the single bond. So the electrons which is present over here, these electrons and these electrons which is present in the polyene electron, they are not belonging to either this carbon or not this carbon, but they are present in the common one, right? So these carbon are like a loose one. In other words, they are orphan electrons, like orphans. Or in other words, they are loner electrons. Always the loner or orphan, they are getting excited whenever they are getting something on that. So here, these electrons now, as soon as they receive the light, if they receive the light, and these electrons are getting, E minus, electrons, they are getting excited. As soon as they get a light, these orphan electrons are polyene. And what I would like to say, these are the electrons which will push the photosynthetic process forward, because they can easily excited. When compared to the other electron which is near to the carbon, which is fixed together, and it is not either belong to this or here, but they are present. So not only one, there are many double bonds structure of the compound, and that is the electron which is being excited. So now you know what is the compound and what is the atom and where these electrons are coming from. So that's what my question is. Okay, so now we have answered these questions. Now we are going to the next stage, and then how the electrons is being excited, what type of energy, in light energy. Okay, they have strong absorption band, light absorption, strong absorption bands in the visible region of the spectrum. That visible region of the spectrum is wave GR. This is the visible region. So whatever the radiations which is coming from this violet color, indigo blue, so these chlorophyll molecule, they can absorb. Okay. So you have to have this radiation, otherwise it is not. So I have a paper saying that, if you want to induce more of a particular product like a starch, you have to have this much amount of light. So if you reduce the light, the plant won't grow. So depending upon the species, that's why Texas certain plants will grow. The same plant which is not growing in the north of Canada or in Finland, where there is no light, not much of light. So you need to have the exposure of light is very important on that. So absorption will be more in the visible region. And as I mentioned before, excited state and ground state, and then they are making the alternate, and then the extra excited electrons, which is releasing the energy. That energy is trapped within the one. So are moved from the one to the other. So the energy is absorbed in the electrons, and the electrons are moving from one shell into the another shell. And this is one molecule, this is another molecule. It will move as soon as it's excited. Okay, and they are called for this phenomena, the charges and everything, as I mentioned earlier, and that is the photo-induced charge separation. Photo-induced charge separation. Photo-induced charge separation. Meaning there's one atom, another atom, another atom. So the several atoms become a molecule, right? And then here the electrons are in paired electrons, but it's been excited when the light is coming up. When the light is there, it is being excited to this from one to here, and then it will return here to the other one. And what happened? It become a charged one because it will have electrons. So it will move the next stage, the next one here, and this become a more of the minus, and this become a charged one. So like this, the electrons is charged moving, when the light is falling onto the leaf or chlorophyll molecule, and the electrons present in the diene or the conjugated diene, that will make a move to the next molecule, next molecule, next molecule, next molecule. And thereby, the electrons being transferred. Okay. And these charge separations, okay, the site of charge separation, where the charge separations, the site of charge separations, they are called the site of charge separations, they are called reaction centers. Reaction centers. And as I mentioned earlier, the reaction centers are where they are accumulating, they are present in the chloroplast, right? That's what we just mentioned earlier. And these electrons are moving, that is used for the NADPH reaction. And you also studied about the bacterial. I'll just walk you through. Bacterial photosynthetic reaction. Photosynthetic reaction. Okay. And the electron transfer in photosynthetic are bacterial reaction centers. So bacteria also, they have the reaction centers. So the reaction centers, now you have a cyclic electron flow in bacteria. Cyclic. It excited over here, and then it is cyclic to over here. So it comes in a cycle. The electrons are moved, and then the excited electron coming back, and then it's feeded again. So this is in bacteria, bacterial photosynthetic reaction, and the reaction centers are cyclic in nature. The electrons are here in the ground state. As soon as the light is coming out, it is excited, and then the electrons are moving, one compound, another compound, and then back onto this one. So it is a cyclic process is going on in a bacterial photosynthetic. But in a leaf, not in a eukaryotic, in a higher species of the plant, you get what? Two photosystems, which I explained earlier. Two photosystems, right? And the two photosystems, what we did study about that, photosystem 1 and photosystem 2. In this, the reaction centers of the photosystem 1 are P680, and then here P is 700. And these are the reaction center, and when the wavelength of the light on this particular wavelength is coming out on the 680, and this photosystem 680, which is present in the reaction center of photosystem 1, and the electrons are excited. And then in the photosystem 2, you get the component of the protein P700 when the electrons rays of the light which is falling in, and these electrons is being excited. So the reaction centers, we call it as a P680 and P700. Okay, they are present in plant. So unlike in bacteria, these electrons are moving and coming back and again and again and again when the it's going a cycle. But here in this one, when the photosystem 2 will constantly supplying the electron to photosystem 1, and thereby it's a progressing. When the light is there, it will move. Another light is there in the rays, and this is being excited and it's moving on. So it is not like a cyclic process, but the progress transfer of electron from photosystem 2 to photosystem 1. As I mentioned earlier, the electrons are used to synthesis, NADPH are reducing power, and they lack more of electrons because their electrons are moved by this light. But this system needs more electrons. That electrons from the water, H2O, water splitting to oxygen, and these electrons are being produced at photosystem 2 and constant supply of electrons to photosystem 1. And thereby, you have a two type of photosystem or reaction centers work together, coordinate together. One system supplied at photosystem 2 supplies electron to photosystem 1, and photosystem 1 is constantly producing of NADPH. And this is being used on the dark reaction, and the photosystem 2 will produce more of oxygen, and the light reaction we use in photosystem 2 and photosystem 1, we use the light energy. In thereby, we are producing more of NADPH in the for the next chapter, substrate molecule. Okay, NADPH. So the next question is the plant's electron flow is not cyclic. Okay, in the plant, electron flow is not cyclic. Where do you find the cyclic? You find only in bacteria, not in the plant. But the electron flow, how electron flow transfers from photosystem 2 to photosystem 1. It's a progress transfer of electron. It is not cyclic. That you should understand. Okay. Meaning, what is that one? Photosystem 2. Okay. I'm just writing the another one. Okay. Photosystem 2, it absorbs the wavelength of light as P680 lambda. Lambda means here the wavelength. 680 is the wavelength of the light. It's the light wavelength. It absorbed this wavelength of light, photosystem 2, and got excited. And then at the time, water molecules, they split. Okay, they split the water molecule, and it will produce more of these electrons, and electrons that will travel this photosystem 2 to photosystem 1. Which I mentioned earlier, the same phenomena. I'm repeating it again and again. Electrons are moving from photosystem 2 to photosystem 1. Electrons are moving. And these electrons move through an intermediate is here, in between that. They are called cytochrome here. I write it here. Cytochrome BF. Cytochrome BF is another compound which is something like an intermediate, which are present over here. When the electrons are moving to photosystem 1, it moves through cytochrome BF. And they are the membrane-bound complex. Is it something like in a respiratory complex 3 in electron transfer chain? The same phenomena, the same type of compound is present here in the photosystem 2 to photosystem 1. And this cytochrome BF, the cytochrome BF, I write it here, cytochrome BF, okay, and this is the intermediate, right? Photosystem 2 to photosystem 1. It gives, this is 1, it's not 2. Photosystem 2 here 2, and here the 1. when it moves, and this cytochrome will produce, it will produce more of proton motive force. This will produce PMF, proton motive force, or gradient. Okay, and where this it will produce, it is in the thylakoid membrane. Thylakoid membrane, which is inside the chloroplast. Thylakoid membrane produce the PMF, and the PMF or the proton motive force will help to produce on the ATP molecule. Okay. Now, the transfer from electrons from photosystem 2 to cytochrome BF, and where the electrons are coming from in the photosystem 2, the supply of the electrons on 2, the compound we call it as the plastoquinone. That is the and generate the proton gradient here. I'll write it separately. I always hear it. Plastoquinone, Q-U-I-N-O-N-E. Plastoquinone, P-L-A-S-T-A-Q-U-I-N-O-N-E. Plastoquinone is the compound which will produce more of electron by absorbing the energy from the light. So plastoquinone supply the electrons to cytochrome BF, and cytochrome BF will produce of a proton motive force, produce ATP, and the cytochrome BF is also present in the thylakoid membrane, and cytochrome BF, and when they transfer these electrons to photosystem 1. So one light energy is captured by the 2 with the molecule plastoquinone, which will generate more of electron, supply the electron through cytochrome, and when they transfer, it will produce a proton motive force that will synthesize ATP. At the same time, the electrons are moving through to photosystem 1, where the next reaction will take place there. So it is something like how the electrons are moving from one compound to the other. So that's what is the magic of this particular chapter. Okay. Now the figure which in your textbook is 19.13, the structure of photosystem 2, and then photosystem electron flow, next figure 14, and 15 is 4 photons required for the 4 flash. So that is very interesting. When when the photons 1, 2, 3, 4 cycle, where when each flash of light, you have 4 flash of light, you get excited, meaning each flash of light, 1 molecules of water is split. So that you can easily monitor by a graphical expression, and that's on 15 in your textbook. A plausible scheme of oxygen evolution from that, and also a proton gradient, cytochrome BF contribution. I mean, these are some of the list of figures which I can walk you through, and also you have several experimental results which will give you an idea. What I'll do, I will go through one of your the PowerPoint over here. Okay. Okay. Here you go. What is this? This is your chlorophyll, chloroplast, and the plant cells. Okay. The plant cells, you have a rigid, like a benzene ring. They are not benzene ring. They are the cell wall. Okay. They are the plant cell wall, and inside that you have a dark pigments around the chloroplast. Okay. This I am getting from your textbook. Okay, all of them. Then these are, if you just recollect what we discussed earlier, like a free energy. That is the energies you can monitor, free energy, which I explained earlier. Whenever the energy is absorbed here by the chloroplast or the photosystem 2, water is split into oxygen. Do you see that? And the electrons are excited, and if it returns to the same molecule, this energy will release as a heat energy. But it is not, but it's going to the next one, that photosystem 1, but the electrons over here, and it is really excited when the another ray of light, but at this photosystem 1, water is not splitting it up, but it is being excited
over, and at the time you also produce of reducing power, and this reducing power is NADPH, the yellow one, and then it generates the proton gradient, and that gradient proton motive force, which you have ADP to ATP synthesis. So this is the light reaction. This is the summary of the chapter what we discussed today. Okay, and we are going to have different aspects now. Light is coming up now. The NADPH. You know, could you see in the light energy when these get excited, how the proton is being produced, proton gradient. You know, the hydrogen molecule from one part of the membrane outside to inside, it is moved. When the electrons going out, because the electrons are getting excited, right? Where the electrons are coming from water molecule. So more of water, and it will produce of oxygen, and these electrons are moving. So the energy, really, the splitting of water splitting energy is coming from the light energy. Light is plastoquinone, and then you get the electrons being transferred here, NADP to NADPH. At the time you get the hydrogen is moving in, if more of hydrogen is here and less of hydrogen, that produces a gradient, and that gradient will, you know, push the hydrogen outside. When it moves outside, this ADP molecule will synthesize to ATP in presence of inorganic phosphate. So you get NADPH produced, and ATP is also produced in the light reaction. Okay, how it happens? Now you can see the structure of chloroplast. Yellow is the outer membrane. The inner membrane here, that's you know, chloroplast inner membrane, and the inner membrane is folding again inside just like a matrix in mitochondria to form, and the inside is the stroma, and in the stroma is filled with thylakoid membrane. This is one thylakoid, another thylakoid, another thylakoid, another. So they are piling of thylakoid membranes here, and they each one pile we call it as a granum. Otherwise, in many we call grana, and one granum, another granum is linked by stroma lamellae. So you have a three space, thylakoid space, the in between the thylakoid membrane there is a space, and then intermembrane space, outer membrane and inner membrane space, and you have a stroma lamellae space. So you have a three membrane and three spaces. And the three spaces are now involved for the electron transfer. That there where you get the reactions are occurring for the electron transport inside the chloroplast molecule. Okay. Now the next one is the electron micrograph. If you see under the electron microscope, the thylakoid membrane looks like this. You see, like thylakoid, thylakoid, thylakoid. And one granum, this is another granum, and they are linked by lamella. So this is the electron transmission electron microscope. Meaning, if you cut out leaf chloroplast, inside the chloroplast you can see this one. This is a 500 nanometer. Imagine that's the length of that thylakoid membrane. And these are the cyanobacteria, and how this bacteria is like a chain of a small one. And these are the single or unicellular organism is being taken up by the large plant in the evolutionary process, and thereby the chloroplasts are entering, or the bacteria is modified into the plant and the endosymbiotic process. Though it has its own reproduction, their own DNA and everything, and the plant nuclears also control over this in the chloroplast. So chloroplast contain nuclear protein coded by the nuclear DNA, as well as the protein encoded by the chloroplast DNA. Both of them are there. So these are the endosymbiotic, how the one microcellular organism is captured by the other organisms. But unfortunately, only plant capture it, and we and the animal, they don't have the chloroplast inside the cells. So if they have the cells, then there is no need to eat anything. So you can just drink carbon dioxide outside, and also you can get more of water. That's it, and you are autotrophs, like, you know. So is it possible? Yes, if it is possible, you can go to space without getting radiations, and you know. So these are the structure of the chlorophyll molecule, magnesium, where the nitrogen, and then you can have the, you know, all the double bond as a polyene, as I mentioned. Also, this is a pentyl ring structure together, which is attached to it as a chlorophyll molecule. Okay. Now, how the transfer? These are the wavelength. Photosystem 2 or photosystem 1. So 700 wavelength. There is a one photosystem exciting occurring. Another one is 680. And so here at the two different range where the excitations of the electrons which is occurring here. Okay. For the two systems. And now you can see the energy at the ground state. When the light is there, it is being excited. One electron, say these electrons is one electron is going in one direction, another one in the opposite direction, so that it maintains the equilibrium. But when the light energy is excited, so what happened? The next one after the excitement, it returns. You see, it returns. Either it returns here and releases the heat, or it can transfer, like electrons transfer from here to here. See, look at here to here. So now you can see the excited molecule D and acceptor molecule A, but because of the transfer, what will happen? This become a positive charge and this become a negative charge on that one. Okay. Now you can see the molecular cartoon, how the molecular structure, non-heme, iron and quinone and bacteriophytin and bacteriochlorophyll, heme molecule, special pair, these are the structure. And then bacteriochlorophyll, the same thing, how the pigment looks like, the chemical structure, biochemical structure. And this again is the bacteria, how the P960 bacteria chlorophyll, when the light is enter here and is being excited, it's a fast electron transfer. Charge separation, this become more of charged molecule, and then they move from here to the here, you can see here to here, and then it's become more of the charged molecule again. From here to here, it moves, molecule is moving, and then hydrogen is getting in, and once the hydrogen is there, and then quinone pool, this is occurring over there. So these are the cyclic process. The same process is going to repeat again and again. This is in bacteria, how the electron transfer is occurring here, cyclic fashion. This is again cyclic 960, excited 960. And the electrons are transferring, cytochrome BC complex and proton gradient that will drive to synthesis of ATP molecule. Okay. Now the light reaction, lambda 480 nanometer, and this is the wavelength, 700 nanometer of light. So 680 light to activate the photosystem 2, 700 nanometer light will activate the photosystem 1, and you will get more of NADPH reduced. And cytochrome B, these electrons are moving from here to here. Water is splitting again. This is again the light reaction in a nutshell, cartoon. And plastoquinone is the one which will push the electron. Electrons are generated from the plastoquinone. Okay. And these are the hydrogen, and they will use this hydrogen to make a proton gradient. And here are the manganese, and this is another cartoon how they are arranging it in the intermembrane structure of these atoms. Okay, the thylakoid lumen and thylakoid membrane stroma and everything. Again, you can see what happened in the photosystem 2. Light energy P60, manganese, water splitting in the manganese atom, and then photophytin, and then plastoquinone, and it's an exchangeable plastoquinone. So it is going in one side to the other. Now let's go to the next one. Flash number. Each time you flash a light. So if you have a 1, 2, 3, 4. So you have a oxygen evolved per flash. How much oxygen is evolved per flash? One flash, oxygen. Another flash, oxygen. Another flash, oxygen. So that means every flash, the light energy is used by the chloroplast to split water into oxygen. So they have measured the oxygen, and then every flash of light. That's the experimental proof. Okay. And you can see that in the plant, how this oxygen is evolved, like in a plant resource. So they have done every flash of light, how much oxygen is released. So these are the centers where the manganese atom is involved in the splitting of water molecule, H2O, and here the H2O, okay. Evolution, electrons are being, because of water, light energy, electron excited, proton is produced. Now you can see the OH and H2O. This hydrogen is removed. Now another electron is moved, and then you get the OH here, H2O, and then if you see here, and the electrons and hydrogens move, so the 2 hydrogen is removed from this particular manganese atom, and then the electron is removed, and now you have another hydrogen which is coming out from here, so OH, and then 2 molecules of water again. It will evolve hydrogen, 1 hydrogen, and 2 oxygen atom or 1 oxygen molecule is released, and it is being synthesized. So this is the way how the water is splitting in manganese atom in the photosystem too, in presence of light energy. Okay. Stroma, thylakoid lumen here, 2 H2O, oxygen, and 4 hydrogen, or the hydrogen. This is the quinone to quinol. Okay. The stroma space. You know what is stroma? The space in between the center of the chloroplast. Okay. And then this is again the same one where oxidation and reduction phenomena. Okay, and how the thylakoid membrane, the iron 4 sulfur clusters, which is involved in the electron transfer. You might have studied even my own research paper, iron sulfur unit cluster, which is responsible for the electron transfer, the same phenomena which is here in the photosynthetic process. Okay. Again, when the iron sulfur centers out the plastoquinone, the plastocyanin, from this, I mentioned earlier, the electrons are being excited from this molecule, plastocyanin, when the light energy, as soon as the light is there, it is being excited to ferredoxin through the P700 system, and then chlorophyll A, and then quinone, and then the 4 iron 4 sulfur units. This is 1 unit, this is another unit of 4 iron 4 sulfur unit, and finally it reaches the ferredoxin. So this is a highly excited state, how the electrons are moving, okay, and different intermediate compound. And these are the structure, how the iron sulfur unit, 1 unit, is how they are arranging it in a cartoon. Okay. So NADPH and ferredoxin. Once it reaches the ferredoxin, it will help in the flavin molecule to synthesize the NADPH. Okay. So that's what it's all about. The electrons moving, and it will produce more of NADPH. You can see the FADH and FADH2, the flavin and the ferredoxin, and then NADP reductase, and these are the coenzyme, and this is the phenomena semiquinone intermediate where the reductase will be produced NADP to NADPH. These are the mechanism, how the oxido reduction pathway. These are overall summary of the photosystem 211. This is a manganese center, water splitting, or 680 rays of light, photon is excited, 680 excited state, and then you have the plastoquinone and the quinone AB and cytochrome BF complex and the PCE, and this is again the photocenter reaction center, or P700 photosystem 1, where another molecule of another flash of light, or another rays of wavelength 700 is excited state, and then A, and then different chlorophyll, and then iron sulfur range, and then you have reductase and NADPH being produced. So how the 2 systems, and then how cytochrome PF complex is involved in electron transport between one photosystem to the another photosystem, then how the NADP reductase with ferredoxin is involved in producing NADPH. These are the reduction of potential they have measured here. Okay. This is one of the experiments. If you see the pH7 and pH4, where the thylakoid membrane, imagine of incubation for several hours, the pH7 and pH, what is pH is the negative logarithm of hydrogen ion concentrations, and then here the hydrogen ion concentration is less, here the more of hydrogen ion concentration. So what will happen according to the osmotic theory, the higher electron level to the lower concentration, it will go. So what will happen? It will, till it will reach the equilibrium, pH4 inside and outside pH4. The incubation for several hours. The equilibrium is managed or maintained here. And then if the rapid exchange of external pH addition of ADP and PI, if you add ADP and PI, what will happen here? More of these, this hydrogen which is present inside the pH, which will go outside, okay? It will go outside. So because of a higher concentration, and then here is the lower concentration because of ATP, and ADP molecule will produce more of, use this hydrogen atom. So what will happen? It will distribute, and again it will go out, and thereby, the outside is pH 8 and the inside is pH 7. So could you see, start with pH 7 inside, and then outside is 4, and then when you add ATP synthesis, you have inside the pH 4 and outside the pH 8. So this is one of the experiment they have done, and this is again the same thing in a different way. Yes.
I have a question on the third one.
Which one? Here?
Yeah, on the third one, when they change the pH
Yes.
outside to 8, do they just add, let's say, the water?
Yeah.
Is the concentration on the outside?
Whenever there is electrons are moving and the water splitting, the proton are be excited, correct? So if a more of because you are adding ADP and PI, that will use the gradient of this gradient, pH gradient across the membrane, use that energy, gradient energy, to convert ATP. If there is no gradient, ADP will never get into ATP. You need a gradient. That's what the chemiosmotic theory in mitochondria, when the electron transport is occurring, the proton are moving from one direction, and thereby, one outer is less and inside is more. Then it will transport across. Then you have a gradient, and that will use. So basically, the mechanism is using the transfer of hydrogen from one side of the membrane to the other. When the hydrogen movement, that is the one where the proton motive force and the ATP molecule require that force. You have to create the gradient. No gradient, no ATP molecule. How the gradient is formed? You need the electron transport. How the electrons is moved? You need the excitation energy. That is from the light energy. So that's one. Is it okay? And this some of the oxidative phosphorylation and photosynthesis comparison. How these electrons are moving, where in photosynthesis NADPH and ATP is produced. But here this is the other way around. Could you see that? ATP is produced outside in thylakoid space from the outside, but here inside the ATP, inside the matrix, hydrogen is flowing in. Could you see that phenomena of how the electrons, here the electrons are pushing out, but here the protons are moving in. Could you see that one here? It's very simple, because photosynthesis and oxidative are directly opposite reactions. But both of them are using the electron transport. One is using it, another one is releasing it. Okay, so that's the beautiful picture scenario. But everything using only electrons and the compounds. But the situation is a different place. Okay, so one side it acts like a villain, and the other side the electrons are acting as a hero. That way, one stage to the other. Stage one is the photosynthesis, another one is the oxidative phosphorylation. So everything is the same one. It is not like a new compound, nothing. But it behaves in a different environment, different centers. The electrons are behaving differently. Okay. So the same one again. So cytochrome BF, how the ATP is being produced from photosystem 1, what happened in the photosystem 1 here. The excitement of the electrons with the light energy. This is again the cyclic, photosystem 700, ferredoxin complex and proton gradient form. And this is the same cartoon which I explained earlier, how the electrons are moving from one to the other. And this is a reaction center. Could you see this reaction center? The light is energy is there, right? And it's like a checkerboard, and this is a reaction center where the electrons are transferred, right? So these are some of the compounds, which is the long chain compound. And they are the compound which is like a carotenoid, lycopene, and those are the compound which is the phytochemical compound, which we are also studying for the medicinal properties of that. But these are the compounds which are long chain, which use the connecting the light energy or conduction of or the excitement of the plastoquinone like that. The excited electrons are transferred, reaching the reaction center. So that's the thing. Chlorophyll molecule B. Lycopene, see, the lycopene is another molecule which is present in the leaves, and they can also do it. And beta carotene, it's also present in the leaf. So that's why if you want to protect your eye, you need to have more of beta carotene, because your pigments in your eye, they need more of beta carotene, carrot, vitamin A, all of them lycopene. I mean, all of them, the compound which involve useful pigment, which will dissolve and getting absorbed and getting into your eye pigment, and they are the one which will help replenish of your old pigments, and with this new one, it will help to synthesize your eye pigment. So that's very important. So natural products. And here the wavelength, how this excitement of 680 and 700 photosystem A and B, how they're coordinating each other. These are this beautiful picture of the X-ray crystallographic or cartoon, how the molecules are arranged in the chlorophyll. How the arrangement here, this is a beautiful, so photosystem 1 is red, green is a photosystem 2. Cytochrome B, F is a yellow, and blue is ATP synthase. Could you see this one here, or the arrangement with the thylakoid membrane? They are the thylakoid membrane, intermembrane space. These are the arrangements of that. Yes, go ahead.
I have a question on the graph before that.
Oh, you want the graph? Okay. Yeah.
That one, I was looking at that, I'm seeing that when you look at the lower level wavelengths,
Yes.
it's like the reverse where photosystem A,
Mm-hmm.
power to A,
Mm-hmm.
absorb at the lower.
Yeah. This one here, right? 400. Yes. This is the wavelength of that. There's excitement is going on that one. And 680 and 700 is the photosystem here. Here also at this particular wavelength, this excitement which is occurring, okay. Here the chlorophyll A molecule is coming out, A here, right? And then the chlorophyll B molecule is absorbed and it is going over there. But the real excitement which is occurring at this wavelength, though the chlorophyll receives 400, the 500 nanometer, it is not the reaction which is occurring. That's why we are saying the photosystem A and B, or the photosystem 1 and 2 will have a specific for that particular reaction centers, the excitement which is occurring. So,
My question is, what's the relationship between the
extinction
extinction coefficient?
coefficient, that's a per molar per centimeter, means how much the electrons are moving, the distance. So they have some measurements over there where the dark reaction, when they flash of light, fraction of the light, and then they will find out the excitement which is coming up. The pen will move up or down. So, and thereby they can find out how much the intensity, the wavelength of light. So they will change, they will put a meter like a colorimeter, you change the wavelength and flash of light, and then see the excitement, okay? And then another turn, 500, what is happening? So slowly they will move. In other words, the automatic machine will slowly, the prism will move one wavelength to another wavelength, and then each wavelength it will flash of light. But they observe only on this particular flash, and 608, 700, 680, and 700 is the one critical, where the reaction is occurring. But here, their excitement is there, but there is no reaction. Meaning, as I mentioned earlier, the excitement is there, but it's falling onto the same level. Meaning, you need large wavelength to activate that one. You got it? So that's why when you see the light is there, but the intensity of light is not enough to produce photosynthesis. The different regional specifics. Okay. Okay. Yeah.
So when you look at the first part, is probably the one that will produce heat,
Yeah.
and
Right.
the one on the left,
Oh, here.
the excitement,
Yes, that's right. That's right.
probably just produce heat.
Yes, that's right. There's no use on that one. Yes, it is not transparent. There's no reaction is occurring. Though, but they have to observe because it absorbs light. So that's what it is giving. Like optical density, in a colorimeter we have done in a class, and all, it absorbs, right? Absorption coefficient. The same thing here, the excitement coefficient. Whatever the OD we call it, the light absorbed and then how much amount of light is absorbed, right? But here we call it the opposite, exciting. So it absorbed, but electrons are exciting. So we are measuring that excitement. And these are the arrangement in thylakoid membrane, and the diuron and atrazine. And these are some of the inhibitors. So certain inhibitors which will inhibit the photosynthetic electron transport chain. They are some of the weed killers. You know, some of them are herbicides. We don't want some of the herbicides, so they want to kill the herbs. That's another good old thing. Like, we don't want to get into the trouble, but some of these are some bad for the environment as well. You know, and these are major groups of photosynthetic prokaryotes: green sulfur bacteria, green non-sulfur, purple, here, and oxygenic and all. So these are the bacteria or the prokaryotes. They expect they may present in another planet like Mars or somewhere else. There's no oxygen is there, but they can still be there. So they are excited over there, but they are purple sulfur or purple non-sulfur. But there's no cyanobacteria that needs water. So we don't know yet. So that will give you more of new dimension. I mean, these are the reduction potential of water photosystem 2, exciting photosystem 1, XY, another excitement, and then NADPH oxidized state. It will go and reduce it. Yeah, another cartoon, the same phenomena. And these are the ATPase activity, percentage of control, the control enzyme, and then the dithiotretol, DTT, the millimolar concentration, and they will produce more of the oxygen. Okay, it will supply, say, modified enzyme, and then modified enzyme and the thioredoxin over there. So here the more of ATPase activity is being activated in the enzyme. ATPase activity is being activated with different concentration of DDT only and DDT thioredoxin. So when the thioredoxin means it absorbs more of electrons, and that will move the ATPase activity to produce more of ATP. So the blue one is a DDT only, and the another one DDT plus thioredoxin, and then here you won't get that much. So thioredoxin it is a control enzyme, and these are the modified enzymes. So don't get confused over here. So what this is another experiment where they did with a different concentration of DDT. It will activate the ATPase activity. Okay, that means it increases the electron flow. Basically, the thioredoxin acceptor, these electron acceptors basically. So if you add an electron acceptor, you get more of electrons being transferred, and it will activate, right? The control enzymes, and though you are adding DDT and also thioredoxin, it is nothing happens because that enzyme is not a modified enzyme. So you can really, by genetical manipulation or biotechnologically, you can change the ATPase activity, a modified enzyme, highly activated one, to produce more of ATPase activity, more of ATP synthesis. So that's the next dimension of biotechnology industries now. Okay, with this we will finish this class. And do you have any questions so far?